All right, everybody, welcome to our presentation. It's me, Christina,
And me, Gage Moose! Era!
and today we're going to be talking about the synthetic opioid fentanyl, how the synthetic opioid fentanyl enhances viral replication in vitro. So, to start, we're going to go over our learning outcomes so you know what to focus on throughout this presentation. First, we want you to recall some facts about the opioid crisis in the United States, to describe the use of ELISA in this experiment, to recall how opioid use affects viral replication, and describe the difference between HCV replication at a low dose of fentanyl compared to a higher dose of fentanyl. And so first we're going to go into some introduction and why this paper is relevant. So the United States is experiencing a major opioid crisis. As we can see, there's a picture. There's virtually no difference between the oxycodone and fentanyl tablets, and so it's creating a lot of unwanted death. And people with opioid use disorder are at significant risk for transmission of injection-associated infections such as HIV, hepatitis B, and hepatitis C. And these drugs of abuse, or certain opioids, have been studied and proven that they promote viral replication and virus-mediated pathology. But fentanyl has not been explored despite its widespread use, and fentanyl is 50 to 100 times more potent than morphine, so it begs the question, are these viral replication and virus-mediated pathologies 50 to 100 times more potent when fentanyl is involved? And so this study evaluates the impact of fentanyl on hepatic viral replication in vitro. Moving on to methods, they evaluated the impact of fentanyl and carfentanil, which is a derivative of fentanyl, using in vitro systems that replicate infectious diseases. So firstly, they used a lot of cell culture, cell lines replicating HBV or HCV at concentrations of 1 nanogram, 100 nanogram, and 10 micrograms. So the cells grew on their own for 24 hours, and then they were incubated with the various drugs at various concentrations for 24 hours as well. After that incubation time, the viral protein that was synthesized was quantified by ELISA. And so ELISA here is answering the question of how much virus grew during the different concentrations of the different drugs. And then apoptosis and cell death were measured by various cell proliferation assays. HCV replicative fitness was evaluated by a luciferase-based system, which is really complicated, so we're not going to get into the nitty-gritty of it. And the genome was sequenced through PCR amplification, and then a lot of fancy software to sequence it, and eventually that sequence that they found was verified by different statistical tests, p-tests and t-tests being some that we would be familiar with.
Alright, so now we are going to talk about results. So, as you can see on the right, we have figure 2 and figure 3. So, the impact of these two synthetic opioids in question that Christina talked about earlier are fentanyl and carfentanil. These were evaluated utilizing in vitro systems that replicated infectious HCV or HBV. So as we see in figure 2, at 24 hours post-drug exposure, low-dose fentanyl had no impact on HCV replication. However, we move to higher doses like 100 nanograms, 10 micrograms, we see significantly enhanced HCV replication. Specifically, it jumps up to 187.6% for those 100 nanograms and 193.8% for the 10 micrograms. Carfentanil, on the other hand, we see increased HCV replication at a low dose of 1 nanogram, but this was not maintained at higher doses compared to the fentanyl. It actually kind of drops off, and so the study doesn't really dive into why exactly that happens, because they were mainly talking about fentanyl. But then we see in figure 3, fentanyl resulted in significantly elevated levels of HCV replication and then in a dose-dependent manner, and we kind of see the steady increase in viral replication with the higher doses of fentanyl. All right, moving on to discussion. Ultimately, drugs of abuse are used to promote viral replication in virus-mediated pathology. Currently, the majority of unintentional overdoses in the United States involve fentanyl or fentanyl analogs. Nonetheless, it is not known how these synthetic opioids may impact viral replication and pathogenesis. So, in studies elsewhere, several distinct opioids promote even HIV replication in immune cells. Potential mechanisms of these studies revealed that it revealed enhanced chemokine receptor expression, inhibition of beta chemokines, antagonism of innate antiviral responses, and altered microRNA expression. All this stuff is nothing you need to know. It's just information that other studies have revealed. Even less is known about the potential impact of opioids on viral hepatitis and liver disease. So this is why this study has been so monumental. Nonetheless, the impact of these synthetic opioids on HCV expression and dysregulation of host cellular pathways has not been evaluated that deeply prior to this current study. In summary, there is an urgent public health need to address the linked opioid and viral hepatitis epidemics, particularly in regions with high prevalence of opioid use disorder and synthetic opioids. Rigorous characterization of the interactions among HCV and its treatments, fentanyl, and host cells will improve clinical management paradigms for difficult to treat populations, as well as facilitate rational public health policies and reveal additional pathways for target-specific therapeutic interventions. So, one more time, going over our learning outcomes. Christina for the first two.
Okay, so what we want you to know, recalling some facts about the opioid crisis in the United States, fentanyl being the most dangerous, as it is 50 to 100 times more potent than morphine, and it gets confused with a lot of other opioids, and so people accidentally take it. And then describing the use of ELISA in this experiment, it is quantifying the protein and measuring the viral protein synthesis within these experiments.
Additionally, for our third learning outcome, we want you to understand that opioid use affects viral replication in a way where it basically just increases it. That's the big picture. It can increase viral replication. Finally, describe the difference between HCV replication at a low dose compared to a high dose of fentanyl. This is basically, can you recall at a low dose of fentanyl what is viral replication levels compared to a high level of fentanyl, high dose of fentanyl? And lastly, this data suggests that synthetic opioids promote viral replication but have distinct effects depending on the drug dose and viral target. As higher viral loads are associated with pathogenesis and virus transmission, additional research is going to be essential to enhance our understanding of opioid virus pathogenesis and for the development of new and optimized treatment strategies. And that, my friends, is the end of the presentation. Ow! Let's have a great semester!